Good morning to all. So I am Dr. Ganesh Kumar. So I am working as Associate Professor in the Microbiology Department, Spiger. So today I am going to take the lecture on the antibiotic resistance. So before that, I would like to introduce about the antibiotics. So recently we have been using the antibiotics for the long time. So since we are using the antibiotic for the long time, so all the kind of bacteria and viruses getting the resistant to the particular antibiotic. Say for example, if you go for the doctor, if you having any fever, cold or any other diseases, they will provide you some antibiotics. They will prescribe you some antibiotics. For example, for the coronavirus, they are prescribing you the azithromycin. So since we are using the antibiotic for a long time, it's all the viruses and microbes getting resistant to the particular antibiotic. This is called the microbial antibiotic resistance. So this microbial antibiotic resistance will be derived from the so many reasons. So why we are using the antibiotics means we are controlling the disease to safe hand, and we are providing the antibiotic to the food additives and to maintain the crops, and for the so many applications we are using the antibiotics. So side effect of the antibiotics will be like nausea and dysentery or so many things. So we have to use the particular concentration of the antibiotics. So if you see the sources of the antibiotics, there are so many things like a wastewater treatment plant and hospitals and agricultures, and there are so many sources of the antibiotics which will increase the concentration of the antibiotics in the environment. So all the kind of bacteria getting the resistant to the antibiotics. So if you see the antibiotic types, there are several types of antibiotics like beta-lactam and aminoglycosides, and there are so many types. So this is because of the mode of action of the antibiotics. So every antibiotics having the different mechanism to kill the bacteria. So basically we are using the antibiotic to kill the bacteria, and so the mode of actions will be different. Like some of the antibiotics, they will kill the bacteria by the cell wall concentration. Some of the antibiotics, they kill the bacteria by inhibit the protein synthesis of the antibiotics. So likewise, there are so many mode of actions of the antibiotics. So you can ask why the bacteria are getting the resistance, because we are using the antibiotics for so many decades. So always the bacteria and viruses are facing the antibiotics. So they are trying to take the antibiotic as a carbon source, as well as they are getting the resistance to the antibiotic. So the antibiotic, that kind of bacteria are getting the resistance, so it's difficult to cure the disease in future days. Say for example, if we are using the antibiotic for a long time, the particular bacteria are getting the resistance. So if after that, if we use the same antibiotics, they won't be any clinical effective, because that bacteria are getting resistance. So we cannot use the antibiotic. So we have to change the another antibiotic for the cure. So you can ask, there are so many ways to get the antibiotic resistance. The main role is the gene transfer between the one bacteria to another bacteria. So if the bacteria facing the antibiotic for the long time, they are getting resistance by means of mutations. After that, the genes getting alterations. So after that, it will transfer from one bacteria to another bacteria. So likewise, the antibiotic resistance getting resistance. So there are several ways for getting this antibiotic resistance. One way is conjugation. So the combination of two bacteria, and they will transfer of the one bacteria to another bacteria, so the resistance will be transferred. And another reason is the bacteriophage. So there are some viruses, they are getting infected by the bacteria, so they can transfer the antibiotic resistance genes into the another bacteria. So you can ask how we propagate this kind of a problem means that's very difficult to control, because antibiotic resistance gene which will be present in the inside of the bacteria and in the presence of the plasmid as well as the DNA. So if the bacteria are getting multiplication, they will transfer to the another places. So here the main problem is antibiotic resistance gene, because gene is only responsible for the transfer of the antibiotic resistance. So likewise, we have to use the different antibiotics. So if the bacteria getting resistant to one antibiotic, we have to use the another antibiotic. For example, if you go for the doctor, they prescribing you one antibiotic means day by day it is getting the resistance. So you have to use the alternative combination of the antibiotic. So it will cure the disease. So this is also the side effect of the antibiotics. So there are several reasons to the antibiotic resistance. One is the main reason is plasmid transposon and the small fragment which is present in the DNA and the genomic islands. So these are all the factors which will make the antibiotic resistance. So there are several techniques we are using in the laboratory to detect the antibiotic resistance, like yeast trap method and the microarray method, as well as the ELISA method. So these are all the methods we can identify whether the bacteria getting resistance or not. Otherwise, if it is a resistant means, so if you give the antibiotic, all the bacteria will be grown around the disc of the antibiotics. If the bacteria is sensitive, so they will not grow on the vertebrate. So this is a determination of the antibiotic resistance. And after that, if you want to confirm the antibiotic resistant genes which is present in the bacteria, you can confirm by the agarose cell electrophoresis. There are so many genes will be responsible for the antibiotic resistance. One of the particular gene is TET gene, and the azithromycin forms THA genes. There are so many genes responsible for the antibiotic resistance. So final summary is, so day by day we are using the antibiotics for a long time. If you are having some diarrhea or cough, any fever, you are going for the doctor. Simultaneously, we are taking the paracetamol, some other antibiotic like azithromycin, clarithromycin, streptomycin. These are all the antibiotics we are using for the cold and fever. So frequently, if you are using the antibiotic, that antibiotic will not be cured by the particular disease. For example, we are using the azithromycin for the coronavirus. So oftenly, if you are using the azithromycin in the future, it will not cure the coronavirus, because the antibiotic will prohibit the stages of the infection. If you are using the frequently, it will not cure the antibiotic disease. So the thing is, we have to use the alternative antibiotic, and you have to use the antibiotic in the separate manner. So these are all the factors will be determine the antibiotic resistance. So the main consequence of the antibiotic resistance is the microbes. The bacteria, virus and other actinomyces will be getting resistance because of the frequent usage of the antibiotic resistance. So in this case, we have to formulate the new antibiotics to kill the bacteria. So these days, day by day, there are so many bacteria getting evolved because of the mutations. This is because of the usage of the antibiotic resistance. If the bacteria getting the antibiotic resistant, they will get evolved, then they will give the another evolution. Say for example, this coronavirus, we are facing this coronavirus for the long decade, but this is not the new, because this is found only in the 2019 because of the evolution of the virus. But this is occur in the so many days. So in this case, we are going to modify the antibiotics to particular epitome of the virus. So this is determined by the antibiotic resistance. So this is a brief example for the antibiotic resistance. So if in the later we can discuss about the long story of the antibiotic resistance. Thank you.